I want you to picture this scene for a second. It's late March, March 2025.
Right.
And there's a 6-year-old girl. We're going to use the pseudonym May, which is what the reports use.
Yeah, that's what the investigative reports call her.
Right. So May is arriving at Xinhua Hospital in Shanghai, and she's rolling a small suitcase behind her.
Like a little kid's travel bag.
Exactly. Inside, it's, exactly what you'd expect a 6-year-old to pack for a fun overnight trip. She's got stuffed animals, Play-Doh, and a tablet completely loaded up with episodes of Peppa Pig.
Oh, wow.
Yeah. She's holding her mom's hand, looking around the sterile lobby, and she is entirely convinced that she is going on a special vacation.
But, I mean, the reality of why she was walking through those hospital doors is, well, it's infinitely more complex and ultimately profoundly tragic.
Yeah.
Because May was about to become the very first person in the world to receive a highly experimental, completely custom-built gene editing therapy. And this therapy was directed precisely at her brain. And 7 days after walking into that hospital with her Peppa Pig tablet, she would be dead.
It's just devastating.
It is. And what's even wilder is that her death would remain totally hidden, kept secret from the public and even the broader scientific community for over a year.
Which is exactly what we're getting into today. Welcome to the deep dive. Our mission today is to unpack this incredibly complex, ethically fraught story that is really sitting at the absolute bleeding edge of modern medicine.
Yeah, we have a lot of ground to cover today.
We do, and just so you know where we're getting all this, we are synthesizing an explosive joint journalistic investigation that just dropped in July 2026, published by Science Magazine and Retraction Watch.
Which was a massive bombshell.
Totally. And we're also pulling from reports from BioWorld, The Straits Times, El Mundo, scientific literature from Nature, and looking at the background of the lead researcher involved here.
So we've got a really robust set of sources to pull from to try and make sense of this.
Right. Now, if you track Chinese biotech at all, your mind is probably already jumping back to 2018.
Oh, yeah. The He Jiankui situation.
Exactly. The scientist who shocked the globe by announcing the birth of the first gene-edited babies. We are looking at a story today where a top Chinese medical university is once again investigating a massive catastrophic incident in an experimental gene editing trial.
But, and this is a really crucial distinction we need to make right up front, setting those two events side by side requires us to understand a fundamental biological difference.
Right. They're not the same thing at all.
Yeah, not at all. The 2018 scandal involved germline editing.
Which means editing embryos, right?
Exactly. Altering embryos to create heritable changes. Changes that pass down to the patient's future children and their children.
Right.
What May received in 2025 was somatic gene therapy.
So meaning it doesn't pass on.
Right. The editing is confined entirely to the cells of a living, breathing patient. It doesn't enter the germline.
Got it.
And somatic gene therapy is this rapidly accelerating field. The goal is to edit DNA or RNA sequences in order to repair or remove defective genes that cause diseases in children and adults.
So this is like the legitimate, heavily pursued frontier, right?
Exactly. This is where billions of dollars in global biotech research are currently flowing. It holds this miraculous potential for curing conditions that we currently consider completely incurable.
And to understand how a 6-year-old girl became the focal point of that frontier, we really have to look at the underlying biology that brought her to that hospital in Shanghai.
Right. Her specific condition.
Yeah. May was diagnosed with Snyder's-Bloch-Campeau syndrome. It is an ultra-rare neurodevelopmental disorder, and it's caused by a pathogenic variant in a single gene called CHD3.
And the function of that CHD3 gene, it takes us deep into the mechanics of epigenetics and chromatin remodeling.
Which sounds incredibly complicated.
It is, but think of it this way. Chromatin is basically the highly condensed complex of DNA and proteins that forms our chromosome.
Okay, the packaging.
Right, the packaging.
Oh.
Now, for a child's brain to develop properly, specific genes need to be turned on or off at highly specific times during growth.
Like a perfectly timed molecular symphony.
Exactly. And that requires the chromatin packaging to be constantly unwound, read by the cell, and then repackaged.
Oh, I see.
CHD3 encodes a protein that acts as an ATP-dependent molecular motor. It literally uses cellular energy to physically slide nucleosomes around and remodel that chromatin structure so genes can be accessed.
Okay, so it's a tiny motor unpacking the DNA. And May had a very specific missense mutation in this gene. The reports list it as CHD3 C.3073CT, PCHARG 1 Euro 25 TRP.
Which is a mouthful.
A huge mouthful. But in practical terms, it means a single nucleotide change. Just a single C swapped out for a T alters an amino acid right in the helicase domain of that protein.
So the motor was malfunctioning.
Exactly. The motor was broken because of one single typo in her DNA, and the cascading effect of that tiny molecular glitch is severe developmental delay.
Yeah, the real-world impact on her life.
Right. By 6 years old, May was slipping further and further behind her peers. She had intellectual disability, her speech was delayed to just very simple sentences, and she had low muscle tone.
I remember reading one specific detail in the reports that really grounded it.
The chopsticks.
Yeah. They specifically noted she was still struggling to eat with training chopsticks. It really paints a picture of where she was at developmentally.
It does, but, and this is a critical point to hold in our minds as we evaluate what happens next, while Snyder's Bloch-Campeau syndrome drastically impacts a patient's quality of life, it is not inherently fatal.
That is so important to emphasize.
May was not in imminent biological danger from her underlying genetic condition. She wasn't dying from this.
Right. The urgency to treat her was driven entirely by the desire to improve her developmental trajectory before the window of early childhood neuroplasticity closed.
They wanted to fix the motor before her brain finished its crucial early growth phases.
Exactly. It was about quality of life, not saving her from immediate death.
And that dramatically alters the risk calculus, doesn't it? When you are proposing a first-in-human, completely unprecedented genetic intervention inside a child's brain.
It absolutely changes the math. You have to weigh the potential improvement against the unknown risks of a totally new technology.
And the scientist who did that math, who proposed this intervention, was Zilong.
And we should establish who he is, because he's not just some fringe researcher.
No, not at all. He is a major heavyweight in the field, a highly prominent neuroscientist affiliated with the Songjiang Research Institute at Shanghai Jiao Tong University School of Medicine.
His pedigree is incredibly impressive. He earned his PhD from the Chinese Academy of Sciences and then did postdoctoral research in the U.S. at UC San Diego.
Under Anya Van Gogh, right?
Yes, a very highly respected figure in neuroscience. So Keyes has this elite training. He goes back to China, builds his lab around the genetics of autism spectrum disorders, and secures immense funding.
And he actually made global headlines a while back, didn't he? Back in 2016.
He did. His team created the world's first genetically modified monkeys that exhibited autistic-like behaviors.
Wait, they gave monkeys autism?
Well, specifically, they modeled Rett syndrome, which is related to autism. They used a viral transfer method to induce mutations in the MECP2 gene in cynomolgus monkeys.
And it worked.
It did. The resulting monkeys showed reduced social interaction, repetitive pacing, abnormal vocalizations. It was a massive, though definitely controversial leap forward for creating animal models of complex human brain diseases.
So he had already proven he could execute incredibly complex genetic engineering in higher-order primates.
Exactly. By 2025, Q was perfectly positioned to push for custom treatments for rare diseases in humans. He was aiming to rival Western institutions and place Chinese biotech at the absolute vanguard globally.
And it was operating in a climate where those custom treatments were just starting to cross the threshold into human reality.
It was a very heady time in biotech.
Yeah, because just a month prior to May's treatment, in early 2025, the Children's Hospital of Philadelphia achieved this massive breakthrough.
Right, the base editing treatment.
Exactly. They used a customized base editing treatment on a baby with a fatal metabolic disorder, and the child survived. Science magazine highlighted it as a Breakthrough of the Year runner-up.
The global race to deploy these bespoke genetic cures was accelerating exponentially. Everyone wanted to be first.
Which brings us to the actual mechanism Q proposed for May, because it leaned heavily on that same cutting-edge approach, base editing.
Right. They completely bypassed traditional CRISPR-Cas9.
Which is the one most people know, right? CRISPR.
Yeah, CRISPR-Cas9 is what most people associate with gene editing. But traditional CRISPR essentially works by breaking both strands of the DNA double helix to knock out or replace a sequence.
Like cutting a wire?
Exactly. But creating double-strand breaks in a living brain carries a significant risk. You can cause unpredictable genetic chaos, large structural variations, or chromosomal truncation.
Which you do not want in a child's brain.
Definitely not.
Yeah.
So instead, Kree's team utilized an adenine base editor, specifically one called T-base.
Okay, so if traditional CRISPR is like using molecular scissors to cut a misspelled word out of a book and pasting a new one in, base editing is what, like taking an eraser to a single typo and penciling in the correct letter.
That is a perfect analogy. It's more of a chemical reaction than a physical cut. It utilizes a disabled version of the Cas9 protein simply as a GPS system.
Just to find the address.
Right, to find the target sequence. Once it parks there, an attached enzyme chemically converts that single mutated DNA letter back to the normal sequence.
So in May's case, it changes that bad T back to a C.
Well, technically, it deaminates an adenine to an inosine, which the cell eventually reads as a guanine. But yes, the end result is it corrects the typo without ever severing the DNA helix.
Okay, the elegance of that on a molecular level is undeniable. It's brilliant.
It drastically reduces the risks associated with double-strand breaks.
But, and this is where I have to push back a bit, how do you actually get this molecular pencil into the brain of a living child? Designing it is one thing, delivering it seems impossible.
And that is the far greater challenge, especially for neurological disorders, because the blood-brain barrier is notoriously impenetrable by design. It's meant to keep things out.
Right. So how did they bypass it?
They had to use a viral vector. Basically, they hollowed out a virus to use as a delivery truck. They chose an adeno-associated virus, specifically AAV9, because it has a known affinity for nervous system tissue.
Okay, so you put the base editor inside the virus and inject it.
Well, here is where it gets incredibly complicated. AAVs have a notoriously small cargo capacity.
Oh, like the truck is too small.
Exactly. A base editor is a massive protein. It is simply too large to fit inside a single AAV capsid.
So what do? You can't use a bigger virus?
They solved it using something called a dual vector system utilizing split endanes.
Okay, you're going to have to unpack that.
So they genetically divided the base editor into two separate halves.
They cut the eraser in half?
Right. And they packaged those halves into two different AAV9 vectors.
Oh.
The concept is that you infuse both vectors into the patient simultaneously. They both have to successfully travel to and infect the exact same target neuron in the brain.
Wait, what? They have to hit the exact same cell?
Yes, and once both halves are inside the cytoplasm of that single cell, they're translated into proteins.
But they're still in two pieces.
Right, but they attach these things called splintines to each half. Think of them like highly specific biological magnets.
Okay.
Once inside the cell, these magnets recognize each other, bind together, automatically splice themselves out, and seamlessly stitch the two halves of the base editor together into a fully functional, full-sized unit.
That is insane. It's honestly like trying to smuggle a massive complex machine across a heavily guarded border. You can't get it through in one piece, so you dismantle it, pack it into two separate crates, and attach microscopic magnetic sensors to the parts. And you send the crates across the border independently, and you just hope they end up in the exact same warehouse. And the moment they are unpacked, the magnets violently snap the machine back together so it can start working.
That's exactly how it works. But remember, this process has to happen not just once. It has to happen millions of times over across the vast architecture of a human brain to achieve a therapeutic threshold.
Because fixing one cell won't cure her, you have to fix millions of them.
Right, and the statistical probability of both viral particles finding the same cell, entering it, and efficiently reassembling the protein, it requires a staggering viral load.
You just have to flood the system.
You have to flood the central nervous system with an astronomical number of AAV capsids just to ensure enough cells receive both halves of the editor.
Which introduces a whole host of logistical and ethical realities, because they weren't creating this therapy for thousands of patients. This was just for May.
This wasn't a drug sitting on a shelf. It had to be custom built from scratch for her specific mutation.
Right. And the timeline detailed in the Science and Retraction Watch investigation outlines exactly how that was financed, because someone had to pay to invent this from scratch.
And this is where the story shifts from just biology to human desperation. In July 2023, May's parents, the reports refer to them by the pseudonyms Jason and Linda, they attended a public lecture given by Qiu.
They were just out there looking for answers for their daughter.
Exactly. And they approached him after the lecture. This initiated an extraordinary financial arrangement.
It really is extraordinary. The family liquidated their personal savings, they gathered contributions from all their extended relatives, and they managed to scrape together approximately 6 million yuan.
Which is roughly 860,000 US dollars.
Nearly $1 million in cash. And that money went directly toward the development of the therapy.
Right. The parents personally funded everything.
Yeah.
The editor design, the preclinical animal testing, the manufacturing of the clinical-grade viral vectors, and the actual hospital implementation itself.
I mean, Is it even ethical for a family to fund their own child's clinical trial? Doesn't that create a massive conflict of interest where no one can objectively say, stop?
That is the core ethical dilemma of what we call N of 1 trials.
Right. A
Wow.
trial with just one patient.
Exactly. The grim reality of rare diseases is that for ultra-rare mutations like May's, traditional pharmaceutical companies won't invest.
Because there's no money in it.
There is no commercial market. The return on investment for a $50 million development process just isn't there for a disease with a dozen patients globally.
So families are left on their own.
Yeah. So patient-funded research isn't inherently illegal. It's a growing reality. But when the same family is the principal funder keeping the lab afloat, the legal decision-maker for the minor, and the source of the sole participant in the trial,
It creates a psychological pressure cooker.
a massive one. For the researchers, there is immense pressure to deliver a tangible result to the exact people paying their salaries. And for the parents, the sunk cost fallacy becomes overwhelming.
You know, if you're listening to this and wondering why on earth a parent wouldn't just halt the process if things looked even a little precarious, you really have to try and put yourself in their shoes.
It's almost impossible to imagine.
You've spent $860,000. You've spent years watching the science progress in the lab. You truly believe you are inches away from curing your child. If a researcher presents data that is even slightly ambiguous, the emotional barrier to withdrawing is insurmountable.
You are completely tapped out, financially and emotionally. After all that hope, walking away just feels impossible.
Which is exactly why the regulatory framework surrounding these trials is supposed to provide an objective, independent firewall, right? To protect desperate families from themselves and from overly ambitious scientists.
Yes, but here is the critical regulatory loophole. This trial proceeded without national regulatory approval from China's equivalent of the FDA.
Wait, really? How is that even allowed?
It was conducted under a provision known as an investigator-initiated trial, or an IIT.
Okay, what is an IIT?
The IIT pathway allows major hospitals to test novel therapies based strictly on the review and approval of the local ethics committee at that specific hospital.
So it bypasses the national government watchdogs entirely.
Yes. And between 2015 and 2023, the number of IITs in China surged 11-fold. There were over 1,000 of these trials happening.
Because they want to move fast.
Exactly. The stated intention is to cut through bureaucratic stagnation and accelerate cutting-edge therapies from the bench to the bedside.
But the downside seems obvious.
The glaring vulnerability is that it places the entire burden of scientific oversight onto local hospital boards.
Right.
You are relying on a local committee, often made up of overworked clinicians, to critically evaluate incredibly dense, unprecedented genomic data.
And often it's data generated by researchers affiliated with their own institution, right, or a closely tied university.
Exactly. It severely dilutes the independence required to safely evaluate a first-in-human viral vector. It's the foxes guarding the henhouse in some ways.
And we see the deadly consequences of that diluted oversight when we look at the preclinical data the committee was actually evaluating, the animal testing.
Right, because every human trial starts with animal models.
So in late 2024, Kree's team achieved what genuinely looked like a massive success in mice. They developed mouse models that had May's exact CHD3 mutation.
And they gave the mice the therapy.
Right. When they delivered the TB base editor via that complex dual AAV system we talked about, it worked. It corrected the mutation, it restored the CHD3 protein levels, and it successfully rescued the behavioral abnormalities in the mice.
And achieving phenotypic rescue in a mouse model is a significant scientific milestone. It's cause for celebration.
But mice are not humans.
No, they are not. Mice have vastly different immune systems and neuroanatomy compared to humans. The leap from a mouse to a human child requires rigorous safety validation in a closer phylogenetic relative.
Which in this case was the cynomolgus monkey.
Right. And this is where the narrative that was presented to the parents severely, tragically diverged from the biological reality that was occurring in the lab.
According to the investigation, the toxicity testing in the non-human primates revealed profound, life-threatening adverse effects.
Yes, the hidden failures.
Out of the 4 monkeys involved in the relevant testing, all 4 demonstrated some degree of liver injury following the administration of the therapy.
All 4.
And furthermore, one of the monkeys reportedly exhibited severe kidney injury.
The mechanism behind this toxicity is really critical to understand here.
Because this is what ultimately explains what happened to May, right?
Exactly. So AVs, the delivery trucks, are generally considered safer than older viral vectors because they don't integrate into the host genome as aggressively. They don't usually cause cancer.
But they still trigger the immune system.
Right. When you administer trillions of AAV capsids, which, remember, was absolutely required here due to that split-and-teen design, the host immune system recognizes those capsids as a massive foreign invasion.
Because the body just sees trillions of viruses suddenly in the bloodstream.
Exactly. And the complement system, which is a primitive part of our innate immune response, can become violently hyperactivated.
So the body starts attacking itself.
Basically. The Kupffer cells in the liver, which act as the body's filtration macrophages, ingest massive amounts of these viral particles. This triggers a cytokine storm.
Just massive systemic inflammation.
Yes, which leads directly to the organ damage observed in those monkeys.
Now wait, if a primate goes into acute organ failure during a toxicology screen, isn't that a hard stop?
Standard pharmacological protocol dictates an immediate halt.
Yeah.
You conduct deeper toxicology screens, you drastically reduce the starting dose, or you scrap it and redesign the capsid entirely.
You do not proceed to inject that exact same payload into a 6-year-old child.
You absolutely do not.
So if the animal data showed severe liver and kidney damage in the monkeys, how on earth did the hospital's ethics committee approve injecting this into May?
That is the core investigative finding regarding this failure, and it centers on information flow.
What do you mean?
It remains murky whether the ethics committee at Jinhua Hospital ever received or critically reviewed the complete, finalized, individual toxicology reports on those primates before granting clinical approval on January 2, 2025.
Are you saying they didn't see the data?
The alarming implication raised by the reports is that researchers might have presented selected averages to the committee.
Oh my God. So if you average the liver enzyme data of 4 monkeys?
You can mathematically mask the fact that one specific monkey experienced severe acute toxicity. It flattens the outliers.
But in personalized medicine, the outlier is the only thing that matters because you are only treating one patient.
Exactly. An N of 1 trial means May could be that outlier.
And what about the parents? What were they told?
The gap between the raw primate data and the information conveyed to the family was immense.
The consent form they signed, specifically the version designed to explain the procedure to a child. I read the translation of it in the sources.
It's heartbreaking.
It read, "Your book has a small mistake, which has caused you to have a disease. Over time, it can get more serious."
It utilized these comforting metaphors about correcting a typo.
It completely focused on the success in the mice. It fundamentally failed to convey that the delivery vehicle required to fix that typo was triggering severe, potentially fatal organ toxicity in the monkeys.
The risks of a fatal immune reaction were just not communicated in a way that would allow for truly informed consent.
And that brings us to late March 2025, the day May arrived with her suitcase.
Right.
She is admitted to Xinhua Hospital. She undergoes the procedure, receiving the intrathecal infusion.
Meaning they injected it directly into the cerebrospinal fluid surrounding her spinal cord to try and get it up into the brain.
Right. And because of that dual vector split-and-teen system we talked about, the dose required to achieve any sort of therapeutic editing involved an immense quantity of AAV9. The preparations contain trillions of viral particles per milliliter.
And almost immediately, within a few days of the infusion, the severe complications began.
May developed a high fever, which, as you mentioned, is the hallmark of a systemic immune response.
Her body was fighting a war.
Then her platelet levels began dropping rapidly. Shortly after that, she experienced complete loss of urine production.
Which is a clear indicator of acute renal failure. Her kidneys were shutting down.
She was rushed into the pediatric intensive care unit, and the Spanish outlet El Mundo spoke to sources close to the family. They detailed this agonizing moment in the ICU.
It's incredibly hard to hear.
It is. Before passing away, May reportedly whispered to her mother, "Mom, I want to go home."
Oh, yeah.
She died exactly 7 days after the injection on March 30th, 2025.
Just 7 days. And this rapid systemic collapse of her organs was later formally classified by the hospital as thrombotic microangiopathy, or TMA.
And the hospital actually linked the TMA directly to the experimental treatment.
They did.
So help me understand this. Why does a genetic therapy aimed at the brain kill a child via kidney failure?
To understand that, we have to look at the pathology of TMA. It starts with severe damage to the endothelial cells.
The lining of the blood vessels.
Exactly, that ultra-thin layer of cells that line the inside of every blood vessel. When those cells are damaged by massive inflammation, the body's coagulation cascade completely panics.
So imagine the body's tiniest plumbing pipes suddenly getting damaged, causing microscopic clots everywhere.
Yes. The body attempts to patch the damaged vessels by forming these microscopic blood clots throughout the entire microvasculature.
And because it's generating millions of tiny clots simultaneously, it uses up all the available platelets in the blood.
Which perfectly explains May's plummeting platelet counts.
Right. And then what happens to the kidneys?
Well, these microscopic clots physically obstruct the smallest blood vessels. The kidneys rely on a vast, intricate network of tiny capillaries to filter waste from the blood.
So when those capillaries become plugged with these microthrombi,
The kidneys simply cannot function. They are physically blocked, they shut down entirely, leading to the loss of urine production and acute failure.
and here is a vital scientific distinction based on the sources, because people might hear gene editing death and think the editing went wrong.
Right. And the experts reviewing this are very clear. Publicly available evidence does not suggest May died from an off-target edit.
It wasn't the base editor accidentally mutating a crucial gene and causing a rapid malignancy or a brain tumor.
No, the molecular pencil wasn't the fatal flaw.
The fatal flaw was the delivery truck.
Almost certainly. The massive payload of trillions of AAV capsids triggered a catastrophic innate immune reaction that attacked the endothelial lining of her own blood vessels.
And the chilling reality here is that this exact mechanism of vascular and organ damage perfectly matches the kidney and liver injuries seen in the primate trials.
It does. The preclinical data predicted the clinical outcome.
The animal models screened that the delivery vehicle was toxic, and they proceeded anyway.
And what happens next is where the story shifts from a devastating, perhaps arrogant medical failure into a systemic effort to cover it up.
The timeline surrounding the publication of Qiu's research is it's baffling.
Right. So Qiu's team originally submitted their manuscript detailing this base editing research to the prestigious journal Nature in December 2024.
Okay, so that was before May had even received the clinical treatment.
Correct. The paper went through the standard, lengthy peer review process, was formally accepted in January 2026, and was published on February 18, 2026.
Wait, February 2026. That publication date is nearly 11 months after May died from the very therapy the paper was describing.
Yes.
And the contents of that Nature paper, the title was, "In Vivo Base Editing of CHD3 Rescues Behavioral Abnormalities in Mice." The things they left out are staggering.
The omissions are glaring. The paper heavily promoted the efficacy of the base editor in correcting the mutation in the mouse models.
Which was true.
It was. And it included data from the non-human primates to bolster the claim that the approach possessed clinical translation potential.
Basically signaling to the world this is safe and ready for human trials.
Right. Yet, the published manuscript completely omitted the severe toxicity data from those very same monkeys.
They just left it out.
Yes. And worse, it made absolutely no mention that a human clinical trial had already been conducted based on this data. It completely hid the fact that the human participant, a 6-year-old girl, had suffered a fatal systemic reaction to the treatment 11 months prior.
And what about the money?
In the mandatory conflict of interest declarations, the authors explicitly stated they had no competing interests.
Despite the family paying $860,000 directly for the research.
They completely failed to disclose that funding, though, interestingly, an earlier preprint version of the paper contained a very vague acknowledgement thanking families for their quote-unquote support.
I have to push back on the whole publishing system here. How can a prestigious journal like Nature publish this? Don't they check if human trials are attached to the preclinical data they're reviewing?
You would think so. But the publication of this paper exposes a massive loophole in how scientific journals compartmentalize data.
How does the loophole work?
Because the authors deliberately framed the manuscript exclusively as a preclinical animal study, the strict technical omission of human data doesn't automatically trigger a violation of baseline publication checklists.
So, because they only claim to be talking about animals, the reviewers only looked at the animal data.
Exactly. Journals primarily evaluate the specific data set submitted to them. The reviewers evaluate the methodology and statistics of the mouse and primate data provided.
But the primate data itself was sanitized. They hid the severe toxicity in the monkeys, which absolutely falls under the purview of reviewing a preclinical study.
Oh, absolutely. The manipulation or omission of the primate toxicity data is a severe breach of scientific integrity, no question.
Right.
But regarding the human data, while the omission might technically slip through a bureaucratic loophole, it profoundly violates the ethical spirit of peer review.
Because context matters.
Exactly. If the peer reviewers and the editorial board at Nature had been aware that a child had died from a catastrophic immune response to the exact dual AAV platform being described in the paper, it would have fundamentally altered how they interpreted the paper's claims about safety and translational feasibility.
It's highly improbable the paper would have passed peer review in that state.
It almost certainly would have been rejected, or at least required massive revisions and warnings.
And this illusion of transparency, this opacity, it extended beyond academic publishing into public trial registries too.
Yes, clinicaltrials.gov.
Right. For those who don't know, clinicaltrials.gov is a global database maintained by the U.S. National Library of Medicine, and its primary purpose is to ensure transparency in human research.
Specifically so, researchers worldwide can track adverse events. If a drug causes liver failure, you want the world to know so another lab doesn't make the exact same fatal mistake.
Right. But the registry record for May's trial, designated NCT 068600672, was last updated on March 6, 2025.
Which was weeks prior to her intrathecal infusion.
According to the investigation, for more than a year following her death, that public database continued to list the trial status as recruiting.
It sat there, publicly signaling that researchers were actively seeking a patient, completely masking the reality that a trial had happened and the sole enrolled patient was deceased.
It was a ghost trial.
Exactly.
And when the hospital's internal mechanisms finally reacted to the fatality, which was in September 2025, 6 months after she died, the institutional slap on the wrist was unbelievable.
It was incredibly muted.
Following an internal review, Xinhua Hospital was fined 24,000 yuan.
That equates to roughly $3,500 US.
A $3,500 fine for failing to properly oversee a trial that killed a child.
And the physician technically listed on the paperwork as the principal investigator, Zilong Qiu, received a verbal reprimand.
A verbal reprimand. And what about the family, the parents who financed the whole thing, who liquidated their savings?
They received zero official institutional compensation. Nothing from the hospital or the university.
Unbelievable.
The journalistic investigation did uncover that one researcher quietly returned roughly $100,000 to the parents out of their own pocket.
Like guilt money.
Perhaps.
Mm-hmm.
But formally, systematically, the family was left to navigate their immense grief and total financial devastation entirely on their own.
The entire ecosystem, from the local ethics board to the international publishing community, just completely failed to halt or adequately report this tragedy.
It wasn't until July 2026 that the silence was finally broken.
Right. When journalists from Science magazine and Retraction Watch finally broke the story with their massive joint investigation.
And the fallout within the scientific community was immediate and severe.
I can imagine.
Independent experts started reviewing the newly revealed primate data. People like Stephen Gray from UT Southwestern, a major expert in the field. He was unequivocal in his assessment.
What'd he say?
He stated plainly that the therapy never should have progressed to a human trial, given the toxicity signals in those monkeys.
And once that was public, the pressure on the institutions mounted rapidly.
Yes. On July 29, 2026, Nature finally appended an editor's note to the published paper.
Basically a red flag on the article.
Exactly, alerting readers to serious concerns about the integrity of the data and confirming an active investigation.
And simultaneously, Xinhua Hospital scrubbed all references to the paper from its official website. They just tried to erase it.
But the international scrutiny was too intense. Shanghai Jiao Tong University was forced to announce a special task force to conduct a comprehensive probe into the researchers' conduct.
And we really need to connect this back to the broader picture for the listener, because this isn't just about one rogue lab. China is actively trying to balance its massive ambition to be a global biotech leader with patient safety.
It's a very delicate tightrope. We saw the regulatory friction during the Hejiankui CRISPR baby scandal in 2018, and we are witnessing it again right now in the realm of somatic editing.
And it is notable that the regulatory apparatus in China was actually shifting just as the story was about to break.
Right. In June 2026, which was just before the journalistic investigation was published, but after the hospital had been internally fined, China implemented revamped regulations governing cell and gene therapy clinical research.
So they were trying to close the loopholes.
Yes, specifically the IIT loopholes. The new regulations restricted these highly experimental trials to a much narrower list of deeply qualified medical institutions.
You can't just get a local ethics board at a small hospital to greenlight a brain injection anymore.
Exactly. And more importantly, the new rules strictly prohibited institutions from charging patients any fees related to clinical research.
Oh, wow. So no more $860,000 entrance fees for desperate parents.
Right. They attempted to sever that direct financial pipeline between desperate families and the researchers evaluating the safety of the therapies.
Which is a huge step.
Yeah,
But the broader ethical dilemma, it definitely still remains.
it does. Joy Zhang, a sociologist at the University of Kent who studies Chinese scientific governance, was quoted in the sources. She discussed this pervasive culture of secrecy.
Because writing a law is one thing, enforcing it is another.
Exactly. There is a persistent gap between the robust safeguards written into national regulations in Beijing and the actual localized implementation of those rules on the ground in individual labs.
And that gap becomes incredibly dangerous in this new era of bespoke medicine.
Because the core lesson here, the synthesis of all this, revolves around how we define safety when we are only treating one person.
Right. An N of one therapy does not justify an N of one safety standard.
The biological reality is that delivering complex genetic machinery like trillions of AAV capsids into the human body carries immense systemic risks. It doesn't matter how precise the editing mechanism itself might be if the delivery truck kills the patient.
And when the patient is a 6-year-old child who cannot provide informed consent,
Right.
when her underlying genetic condition, while severe, is not immediately life-threatening,
Yes.
and when the family is bearing the entire financial burden of the research, the threshold for independent oversight must be absolute. It cannot be optional.
The parents in this case were literally purchasing hope. But the complex, severe risks of the viral delivery mechanism were inadequately communicated to them. It fundamentally compromised the integrity of their consent.
As we move further into this era where bespoke, custom-coded genetic cures are theoretically possible, the medical community really has to establish rigorous, globally enforced standards for how these therapies are developed, funded, and reviewed.
We possess the molecular tools to edit the fundamental code of human life right now. But our ethical and regulatory frameworks are desperately struggling to keep pace with the biology.
Which brings us to the final thought we want to leave you with today.
Yeah.
We've spent this time breaking down the chromatin remodeling, the split and pain delivery systems, the institutional cover-ups, and the shifting regulations. But beneath all the complex biology and biotech policy is just a really raw, devastating reality of parents trying to save their child.
The psychological weight placed on the parents of children with ultra-rare progressive diseases. It's unimaginable.
They are told by the traditional pharmaceutical apparatus that their child's life does not represent a viable return on investment.
So they're left entirely to their own devices in a rapidly advancing, very dangerous scientific landscape.
And so we have to ask, consider the ecosystem we are building. In an era where bespoke medicine is possible, are we inadvertently turning desperate parents into venture capitalists gambling with their own children's lives?
It's a terrifying thought.
Are we placing them in a position where they have to critically evaluate complex toxicology reports that even local ethics boards fail to comprehend? If science can now build a cure from scratch for just one specific person, how do we protect that one person from becoming just another acceptable data point in the relentless pursuit of progress?
It's a question that demands an answer before the next bespoke therapy moves from a primate lab into a pediatric ICU.
It really does. Thank you for joining us on this deep dive. Keep questioning the headlines, keep looking beneath the surface of the next miracle cure you read about, and we will see you next time.